Hello scholars, today we are going to complete our series on microbial physiology. In the first video we talked about PR fermentation and MRVP. In the second video we discussed the urease test, BEA, citrate, decarboxylase, and PDA. Today we are going to end with the catalase test, oxidase test, the SIM, and nitrate. Let's get started with the catalase test. Catalase is an enzyme and it breaks down hydrogen peroxide. Hydrogen peroxide is extremely toxic to cells and it wreaks havoc on the cell. Hopefully you can remember that in the lecture we discussed the different toxic forms of oxygen and hydrogen peroxide was one of those. However, if a bacterium can make catalase, the enzyme that breaks down hydrogen peroxide, then it will be able to survive in the presence of that toxic form of oxygen. This is the equation. Hydrogen peroxide, which is the substrate, is broken down into water and oxygen if catalase is present. So in order to test for organisms that are catalase positive in our lab, we will simply use the substrate hydrogen peroxide as the reagent. There are different ways to conduct this test in the lab. First, you can simply purposely grow up a slant of your organism and pour the hydrogen peroxide directly into the slant after the organism has been allowed to grow. The downside of this, however, is that if you were planning on reusing the organism, you wouldn't be able to. So it is always best to grow this up ahead of time if you want to use this method, because once you use it, then you must dispose of the tubes. However, you can grow up the organism, and if you want to use the organism for other tests, the way that you can save that stock is you can dip something like a wooden stick into the organism and then take the tip of the stick and submerge it and dip it into the hydrogen peroxide as you see in this particular method. Another way that this can be done is by taking a bit of the organism and placing it on a slide and then adding hydrogen peroxide to the organism on the slide. If you use this method in the lab, I strongly suggest that you cover it with a petri dish in order to prevent any splattering that can occur from a positive result. Now, as stated earlier, the hydrogen peroxide, if catalase is present, is broken down into water and oxygen. The oxygen is evidenced by bubbles. If you see bubbles, then that means that oxygen is there and the hydrogen peroxide is being broken down by the catalase. Therefore, a catalase positive organism will yield bubbles when the hydrogen peroxide is added to it. If the organism is negative for catalase, there will be no bubbles. This is a very quick test to perform in the lab. It yields instant result, and it is used to identify organisms that produce this particular enzyme, catalase. It helps us differentiate catalase positive Micrococcus and Staphylococcus from the catalase negative Streptococcus and Enterococcus and Lactococcus genera. The next test on our list is the oxidase test. Many bacteria have electron transport chains that resemble mitochondrial electron transport chains in eukaryotic cells. The electron transport chain is embedded within the membrane. This picture is showing the membrane of the cell, and the purple structures that you see embedded within them are proteins. This chain consists of 4 large enzymes or proteins, and these proteins are enzymes in this case, that we broadly name complex 1, 2, 3, and 4. Each of these contains molecules jointly able to transfer electrons down towards oxygen. The last enzyme in this chain is complex 4. And we call that cytochrome C oxidase because it makes the final electron transfer of the chain from cytochrome C, circled in blue, to oxygen, reducing oxygen to water. The oxidase test is designed to identify the presence of cytochrome C oxidase. It is able to do this because cytochrome C oxidase has this unique ability to not only oxidize cytochrome C, but to catalyze the reduction of cytochrome C by a reducing agent. It's a chromogenic reducing agent. This agent is called tetramethyl P phenylenediamine. You don't have to remember that name. But this agent is a reducing agent and it will develop color if it becomes oxidized. The oxidase test is performed in various ways. The reducing agent can be added directly to bacterial growth on the solid media, but this is not the most convenient way for it to be done. There is also a commercial test system with the reagent already on the slide, which is shown here, and a bit of the organism can be applied to the slide. Alternatively, a small amount of growth can be transferred to a piece of filter paper and simply add the reagent to that. And the one that we most commonly use in our lab are the strips, which already come with the reducing agent within it. All of these tests must be read within 20 seconds. If the organism is oxidase positive, then it will turn to a bluish or purple color. If it is negative, you would see sort of a yellow staining, but we just call that no change. This test must be read within 20 seconds, so you must be very precise when adding the reagent to the organism or adding the organism to the dry slide or the strip. If color change comes at 21 seconds, then this is considered negative, and this means that the organism does not contain the enzyme cytochrome C oxidase. Our third test that we're looking at today is the SIM. SIM is actually a combination test because we are checking for 3 different activities of the cell. One is sulfur reduction, which is where the S comes in. The other is indole, which is what the I represents. And lastly, motility, which is what the M represents. This particular medium includes casein and animal tissue, which are sources of amino acids. There is also an iron-containing compound called ferrous ammonium sulfate, and then there is sulfur in the form of sodium thiosulfate. This medium also has a reduced agar concentration. Typically, agar concentration is about 1.5% in the nutrient agar, but for this particular medium, it's 0.35%. You would see the significance of that when we talk about motility. The first part of the SIM is the sulfur reduction. Now remember, this particular medium has a source of iron in the form of ferrous ammonium sulfate. And of course, it has the sulfur in the form of sodium thiosulfate. The S part of the SIM is looking for sulfur reduction to hydrogen sulfide. This can happen in 2 ways, and it all depends on which enzymes the organism possesses. If the organism possesses cysteine desulfurase, then this enzyme will be able to hydrolyze the amino acid cysteine into pyruvate, but you would also see that this results in the formation of hydrogen sulfide, which is a gas. If the organism possesses thiosulfate reductase, then it will be able to catalyze the reduction of sulfur in the form of sulfate to hydrogen sulfide. Both of these processes result in the production of hydrogen sulfide. Now remember, this medium contains iron in the form of ferrous ammonium sulfate. When the hydrogen sulfide, which is a colorless gas, is produced, it will be detected in this medium because it reacts with the ferrous sulfate in the medium, which again is the source of iron. And it will produce a black precipitate called ferric sulfide. Therefore, the results here would be positive if the organism produces this black precipitate. If there is no change, then it would be considered negative. In the tube shown here, this first tube is negative because there is no black precipitate, so it is negative for sulfur reduction. And the second tube is positive because of the black precipitate. The next portion of the SIM is indole. The medium ingredients that come into play here would be the casein and the animal tissue, because this is the source of the amino acid tryptophan. Bacteria that possess the enzyme tryptophanase can hydrolyze the tryptophan into pyruvate and ammonia, as well as indole. This particular test is looking for the production of indole from that tryptophan. The way that we check for this is after the incubation, we will add a reagent called Kovacs reagent. Kovacs reagent contains p-dimethylamino-benzaldehyde. We simply call it DMABA. There is also some hydrochloric acid dissolved in amyl alcohol. When a few drops of the Kovacs reagent is added to the tube, it will form a layer over the agar. The DMABA in the Kovacs reagent will react with the indole, if it is present, and it will produce a quinoidal compound that turns the reagent layer red. Therefore, a red color in the alcohol layer will indicate a positive result, whereas no color change will indicate a negative result. Now we're going to discuss the last part of the SIM, the motility portion. And determining motility in the SIM medium is made possible because of the reduced agar concentration. In normal media, like the nutrient agar, for example, the concentration is about 1.5%. But in the SIM, it's down to 0.35%. So it's not very dense. Also, the method of inoculation of the SIM is what allows us to determine motility. For the SIM, we use a needle. And what we do is once the needle is incinerated and you pick up microorganism, you will take the uninoculated tube and go straight down with the needle and right back up along that very same stab line. If the microorganism is motile, meaning that it possesses flagella and it's able to move about, it will not stay in the stab line. It will move outwards and radiate outward from the stab line. However, if the organism is not motile, it will remain in the stab line and grow there alone. Looking at the results here, you would see that in this very first tube, let's call it A, you are not able to see that distinct stab line anymore. And this is because the organism left the stab line and moved outward into the medium. However, in tube B, you can see the very dense growth within the stab line. It did not move outward. This means that the organism is not motile. So a positive motility test for the SIM would look like tube A, where the growth in the stab line is not evident, but a negative result will show the growth remaining within the stab line itself. So this wraps up all of the tests that we get out of SIM. It's a combination test, meaning that we are able to check for sulfur reduction, indole production from tryptophan, and motility. Now let's get some practice with being able to read different types of results that we can get from the SIM. In tube A, we don't see black precipitate. This means that in tube A, it is negative for sulfur reduction. However, we do see the red layer, so that means it's positive for indole. And for the motility, it is also positive because even though we see a slight stab line here, we do observe that it's very cloudy in the surrounding areas. So this means that the organism moved out of the stab line. So in this case, the result will be negative, positive, positive for the SIM. This organism is E. coli, so it is motile, it does not reduce sulfur, and there is indole production. Now let's take a look at the results in B. This is Staphylococcus aureus actually, and there is no black precipitate. Therefore, it is negative for the sulfur reduction. Also, there is no red layer, so it's negative for indole production. Motility is also negative because, as you will observe, the stab line has all of the growth within it. It's very dense and there's no radiation of the growth outside of that stab line. Now let's look at tube C. Tube C is S. arizonae, and this shows one of the problems that someone can often encounter when performing the SIM test, when reading it. Now, this is clearly positive for sulfur reduction, and there is no indole production, no red layer. But it's very difficult to perceive the cloudiness from the motility. For this example, you do see some cloudiness at the very top where the black precipitate does not obscure the motility. So this one is scored as a positive in motility. If you were in the lab, it probably would be much easier for you to see this in person. Now let's look at D. For D, we have E. aerogenes. There's no black precipitate, so it is negative. There is no red layer, so it's negative for indole as well. And for motility, this one has moved outside of the stab line. This would be positive for motility. So the result for this SIM would be negative, negative, positive. Lastly, we have E. This organism is P. vulgaris. It's positive because it's red, and you can see the cloudiness throughout the medium that radiated out of the stab line. So it's also positive for motility. We get a lot of information from the SIM test. And please keep in mind that each one of these would have been inoculated with a needle right down the middle, and the needle would have been carefully removed right along the stab line. Now we're finally on our last test, the nitrate reduction test. The medium that we use here is nitrate broth, and the ingredients include beef extract and peptone for nutrients. And because we're looking to see if the organism is able to reduce nitrate, then we have to add a source of nitrate. In this case, it is potassium nitrate. One way to check for the utilization of nitrate is if there is a gas produced. So we include the Durham tube to see if any gas is going to be trapped in the tube. That should sound familiar to you because in the PR fermentation test, we had the Durham tube to see if any gas was produced, if the fermentation of the particular sugar took place. In chapter 5, in our lecture on metabolism, we spent a great deal of time discussing aerobic respiration, where electrons were passed off to the final electron acceptor, which was oxygen. But we also learned about anaerobic respiration, and this involves the reduction of an inorganic molecule other than oxygen. Nitrate reduction is one of those examples. Many gram-negative bacteria, including most Enterobacteriaceae, contain the enzyme nitrate reductase. If they contain this enzyme, then they are able to perform a single-step reduction of nitrate to nitrite. Then there are other bacteria in a multi-step process that's called denitrification, are able to convert nitrate to molecular nitrogen or nitrous oxide. In this figure, we show some products of nitrate reduction. Nitrate reduction can be quite complex, and many different organisms under different circumstances perform nitrate reduction with different outcomes. Here are some of the outcomes. Some can simply, in a one-step process, take the nitrate and reduce it to nitrite. So that's one of the products that we can get. Other bacteria that we call denitrifiers are able to take the nitrate and reduce it all of the way to nitrogen gas. Both of these processes would be considered anaerobic respiration. Then there's some organisms that are capable of what we call assimilatory nitrate reduction, where the nitrate or the nitrite is reduced to NH4. And the NH4, which is ammonium, can then be used to construct amino acids. From this figure, we can deduce that reduction products are nitrite, nitrogen gas, also ammonia. These are a few of the possible end products of nitrate reduction. When reading the results of this test, it is not as straightforward as the other tests that we have done, because there are 3 ways in which this test can be positive. Let's remind ourselves of the reduction products: nitrogen gas, nitrite, and ammonia. Let's say these 5 tubes were ones that you inoculated in the lab. Tube number 2 is uninoculated, because we want to have a negative control to compare our results to. The first way that this test can be positive is if the organism produced gas. This is denitrification, and it's a visual test for the presence of nitrogen gas. All you need to do is look at the tubes. If you see bubbles trapped in the Durham tube, then it may, and notice I said it may be positive. In tube number 5, we see some bubbles. As a matter of fact, there's so many bubbles that you cannot even really observe the Durham tube very well. Now, just because you see bubbles is not an automatic positive for nitrate reduction. In fact, if you see bubbles, then you also need to compare it against the PR fermentation test. Recall that the PR fermentation included broth such as the PR sucrose, PR glucose, PR mannitol, etc. When you compare your gas result in the nitrate reduction to the fermentation tubes, if your fermentation tube is negative, that means that now we can score your nitrate reduction test as positive, because we know that the gas is from the nitrate if your fermentation was negative. But if you have a bubble in the nitrate and then you look at your results from the phenol red test, if that has a bubble, then we cannot say that it was positive. This is undeterminative, because the gas that we see may or may not be from nitrogen. Now remember, I said there are 3 ways that this test can be positive. This is just one way that the test can be positive: having a bubble in the nitrate test and not having one in the PR fermentation. If there is a bubble in the PR fermentation, then we cannot determine if the gas that we see in our nitrate is from nitrate or is it from another source. So now we have to move on to the second test with these tubes. Now, just for explanation purposes, we're going to imagine that tube number 5 has bubbles here in the nitrate test, but it does not have it in a PR when we compared. That means that tube number 5 should be considered positive at this point. However, tubes 1, 3, and 4 do not have any visual evidence of denitrification. So we're going to have to move it to a second phase of testing. Now, we are going to ask the question, is the nitrate then reduced to the nitrite? We know that it does not have gas in 1, 3, and 4, but we still can't mark it negative. Actually, very few organisms make gas. So the next question here is nitrate reduced to nitrite? This is quite simple to figure out because all we have to do is test for nitrite. So tubes 1, 3, and 4 will all get reagents A and B added to it. And if the color change is not immediate, we're going to wait 10 minutes. Here are your results. As you can see, tube 1 turned red. Therefore, nitrite was present. That means that this organism reduced nitrate to nitrite. So we are going to score number 1 as positive. We've already determined number 5 is positive because of the gas, which leaves us now with tubes 3 and 4. Even if there is no change, we still need to do one more test. Why? For number 1, we checked for nitrogen gas. Number 2 here, we checked for nitrite. But remember, there are other reduction products that we have not checked for, like ammonia. So tubes 3 and 4 may very well be positive. How do we know that the nitrate wasn't turned into something else? This is where phase 3 comes in. Is the nitrate reduced to other compounds? Now, we have checked for nitrogen gas, we have checked for nitrite, but another consideration is that the organism used the nitrate and just didn't turn it into nitrogen gas or nitrite. So how do we determine if the organism used the nitrate? Well, let's think about it. How would you determine if your kid ate the broccoli off of their plate? You would look for the broccoli. If the broccoli is there, then they did not eat the broccoli. But if the broccoli is not there, then they used it. Now, using that example, let's think of the nitrate. Let's test for it. If the nitrate is there, then that means that they did not use it. If the nitrate is not there, then that means it used it. So we're going to test for the nitrate. How do we do that? We would add zinc powder to tubes 2, 3, and 4. We're going to add it to tube 2 because remember that was uninoculated, so it should be negative. We're going to use that as a negative control. Now this is where the twist happens. In situation number 2, a red color was positive. This means that nitrite was there. But now we're going to check for nitrate. If it's there, this would be negative because the organism didn't use it. How do we check for it? We add zinc powder. When zinc powder interacts with the nitrate, if it's there, then it will turn red. In this case, red would be negative because that means that nitrate is still there. It was not used. However, if there is no change, that means that there is no nitrate there for the zinc powder to interact with. This means that the organism is positive for nitrate reduction. Let's take a look at the resulting pictures when we add zinc to tubes 2, 3, and 4. Tube 2 turned red. Well, it should, because remember, tube 2 is a negative control. It was uninoculated. That means nitrate is there still. Nothing was there to use it. So that nitrate interacted with the zinc powder and turned red. Tube 3 also turned red, and that's because the nitrate was there. When we compare it to the uninoculated control, they both look the same. Therefore, these organisms would be scored as negative for both. Now, for tube 4, we added the zinc powder, and there was no change. This means that there was no nitrate there to react with the zinc powder. Therefore, the organism in tube 4 used the nitrate. It's no longer there. So this would be considered positive. Now, no color change after the addition of zinc simply indicates that the organism reduced the nitrate to ammonium, nitrous oxide, nitric oxide, or just something else that we did not test for, or some non-gaseous nitrogenous compound other than nitrite that we weren't able to test for. So now we have wrapped up all of the tests that we typically use in our lab under the umbrella of microbial physiology. We've covered the P-off fermentation, MRVP urease, BEA citrate, decarboxylase, PDA catalase, oxidase, SIM, and nitrate. These are the physiology tests that we use in the lab to aid us in identifying microorganisms. If there is anything that's left unclear to you, please feel free to contact me so that I can get your questions answered. In the meantime, happy studying.